Good afternoon. Welcome to everybody who's joining us this afternoon for the EPI Town Hall. Again, we thought we'd do this to sort of draw the year to a close. It was just a few weeks after this time period, back a year ago in February of 2020, that the first SARS-CoV-2 isolate was identified here in Gainesville. It's been quite a year in the intervening years. We've done a lot of work with SARS-CoV-2 and COVID. For those of you who are not directly involved with COVID research, you were directly impacted because labs were shut down. Things are starting to return to normal at this point, although we still have a way to go. And so what we wanted to do today was to kind of run through a brief summary of all the stuff that's been going on for the past year, particularly since a lot of people have been not physically present at EPI, to talk about some of the findings that have come out of the Institute, and to talk about where we're headed from here. What does the coming year hold? What have we got scheduled? What's planned? Where do we go from this particular point in time? So we've got a number of presenters today. If I could have the first slide. And again, my thanks to everybody who tossed in a few slides into the discussion for this morning. I would note there are some folks who can't be with us today. In particular, Roald Dinglinson had a conflict with a class and couldn't join us. So we're going to miss hearing from his lab. And I know there are others who we may well have missed, but hopefully we'll give you a feel for some of the range of activities that have gone on within EPI within the past year. So first slide. Next slide. To summarize or to provide an agenda for the afternoon, objectives are to review where we stand and what EPI faculty have done more than a year, well, actually just barely within a year of emergence of SARS-CoV-2 in this region, and to explore directions of the future. We're going to talk about the past and current SARS-CoV-2 research activities, initial testing programs, other research, including current projects. Mike Lauzardo is going to talk some about the Test, Trace, and Protect program, which much of which is housed here, and then talk a little bit about where things stand overall. Next slide. So I think the first thing to note is that investigators here at EPI have been engaged with coronaviruses for a lot of time before COVID showed up. John Lednecki in particular has done a great deal of work with SARS-CoV-2. This is actually a paper that he put out back in 2017. Had a grad student go over, collect feces from the bat houses here on campus, and there was coronavirus. Not the coronavirus that we know today as SARS-CoV-2, but a coronavirus. And actually, because of this, when we did start seeing cases of COVID beginning to appear in the United States, investigators here at EPI were uniquely positioned to be able to rapidly develop PCR and other diagnostic techniques. And I would note it, with John, we've also been working with some of the endemic human coronaviruses, which cause common cold symptoms in Haiti. I know Derek Cummings and his group have been involved with the endemic human coronaviruses for a number of years. And I think he's going to talk a little bit in his section of this presentation about some of the work they've been doing. But again, to emphasize that coronavirus was not a total unknown when it suddenly appeared on the landscape here. Next slide. So when we first started getting word about cases appearing here in the United States, if you recall back a year ago, the issues with diagnostics were significant. The only diagnostics available were being done at CDC. CDC actually was telling people not to do their own diagnostics because they wanted to try to keep everything at CDC and then subsequently at the state labs. So we decided that as the role of the EPI was to get diagnostics up and running. And so building on the work that John Lednicki had done with PCR, we set up assays. We converted one of the Plex labs to a BSL-2+ facility for RNA extraction. I did some arm twisting with Adrian Tyndall, Dean of the School of Medicine, College of Medicine, and we were able to get the necessary robots. We had an assay up and running, and we're screening individuals here in town. We were the only game in town. It was not being done by Shands or by UF Health. And actually, we identified the first clinical case at Shands on March 10th, 2020, designated as strain UF1 in our strain collection. Subsequently, obviously, there have been a number of other resources that have been brought to bear. And as Mike Lauzardo will talk about, nowadays, running a couple of thousand assays in a single day is not that big a deal. But at least for us, we were here at the very beginning when there was no other way to get the diagnostics done. With this high throughput laboratory, which we set up here at EPI, we have processed over 8,700 samples to date. Most of that was back at the beginning, but we have continued to run samples here as part of research protocols. Next slide. And the key thing that allowed us to set up this high throughput system was the volunteer labor. And I really want to say thanks to everybody who participated as part of the high throughput PCR laboratory that we had here at EPI. Again, this was a situation where many of the other labs, if not most of the other labs on campus, were shut down because of concerns about COVID. And so individuals who were normally employed by those labs were able to volunteer and work with us. And as you can see, there were a number of individuals that played a critical role in terms of our ability to do the number of assays that we were able to do with basically as a research lab, but serving again as a key component of the overall approach to diagnosis, early diagnosis of SARS-CoV-2. Next slide. And as we got underway, as I said, our initial focus was on the identification of, or the setting up a lab that could do PCR assays for SARS-CoV-2, but it rapidly became apparent that we also needed the ability to do serologic screening. And actually, I'm going to ask Mo Long to talk a little bit about some of the work that she did in setting up sort of a parallel serology laboratory that was also capable of handling a substantial number of samples. Mo?
Hi. So early on, Glenn called me and said, "Could you develop an ELISA for SARS-CoV-2?" And I said, "Sure." And that was really the shortest thing I ever did in terms of saying yes. And this has been kind of the longest thing. But at that time, there was some commercial ELISAs and rapid testing for antibody, but you could not get them. They're still rather expensive, in my opinion, and they really did not want them to go to research laboratories. And then some of these have been developed with very assay-specific equipment. And then as I don't know if you follow the literature, but the sensitivity, specificity, and really the agreement across tests is highly variable. And so at 14 days post-infection, one of the studies, which was done actually in August, and there's newer studies, but still, there was variation in terms of sensitivity across seven different tests and different formats, and the global market leader was actually the lowest. So some of the first ones on the market used things like the nucleoprotein and the spike protein, and they don't really do a good job in terms of specificity. They may be sensitive, but they really don't differentiate background exposure too well. The lateral flow assays, which to me, the best use would be as a rapid screening, both in the house and say for kids walking into school, they even run faster in terms of their timing. And so they have really poor agreement, and the confidence intervals are really large. And so at that point, Eric Nelson and his colleagues at Harvard pretty much saved us, because we were looking at using a whole virus assay, which would have been really cumbersome and require a lot of growth of the virus at BSL-3, which was not attractive. And so they basically gave us the plasmids for both the monoclonal anti-SARS humanized monoclonal, and also the receptor binding domain, and enough to get started. In fact, scads of protein to get started on the ELISA. Next slide. The person in the picture, by the way, I don't have as big of a team. There are a lot of people on the ELISA, on the PCR team that were also helping on the ELISA, but right there in the middle is Dr. Rayu Pu, who's working at the bench, and I have to give about 99% of the credit to, and he pretty much does most of the work, and with a little bit of help from OPS, but he's really been amazing in developing all the assays and running most of the samples. So next slide. So I'm just going to show you a little bit about test performance, because there's a lot that people are going to show, and their samples are our samples. So I think the PCR is up around 8,000. I think last time I counted, we were getting close to 2,500, or about 2,500 to just below 3,000 samples, and about 2/3 of them we run IgG, IgM, and IgA on. And so this is just some of the analysis that Derek Cummings has done, and basically on your left is the ODs, the raw ODs, looking at pre-pandemic controls, which are the black dots, and then the PCR positive post-pandemic onset at 4 weeks. And so these are our attempts to set a cutoff, and there's a fairly, some of these are early on, more like 2.5 to 3 weeks. Most of them are around 4 weeks. So we get some overlap, but as you can see, the negatives to the RBD really spread out along the bottom distribution. And then we also do an immune response calculation, where we actually run all of our samples against BSA, because I wanted to know, kind of on a calibration standpoint, what the individual response was to non-specific proteins. And so I really pushed this and thought this would be much better, but actually the raw OD gives us a little better discrimination. Next slide. And so this is the receiver operator curves generated for sensitivity and specificity at 4 weeks. For those of you that know about the receiver operator, it's comparing the sensitivity, specificity for a given sample, and basically the area under the curve, it should be a nice square. If you look at the top figure, and you can see we come to almost not a perfect square, but pretty close. And so at 4 weeks, we have a 99% confidence that that on a positive PCR or PCR positive person, they're going to have IgG. So this is just for IgG. And with this, at the optimal cutoffs we've been setting now, we can bring the sensitivity up to 100% and specificity at 98.6 in individuals that have that are 4 weeks out from infection. Once you get a little closer, obviously you lose your sensitivity, and then you can see there's kind of a tight cutoff between the positives and negatives. We can, with Derek's analyses, we can kind of play around with this and look really why some of these people sit in the middle versus people that basically are negative and want to sit a little higher than they should. So And the next slide, I think, is my last slide. And so these are the changes over time. And on your left is the IgG, in your middle is the IgA, and on your right is the IgM. And basically, IgG and IgA rise at the same time post-infection, with the IgA decaying quicker over time, though the IgG continues to decay and is slower. The weeks post-infection is down on the bottom. We now have several people we've gotten at 6 months, and they have a pretty good, still pretty good, robust IgG, and some have a pretty good IgA response. The IgM is much lower, probably much less of a reliable test for acute exposure, but this is an indirect ELISA. No one has done an IgM capture, and that is where we're going with the IgM to see if we can get a little better sensitivity on the IgM. The next assay we're developing is animal testing. We've got a little bit of money for the state, and I'm really interested in reverse reservoiring. And then also we are going to look at other proteins to SARS to do comparative ELISAs to see if we can look at vaccine versus natural infection using ELISAs, and maybe coming up with some sort of index comparing the other proteins. And that's pretty much all I have.
Thanks. You know, we've, as should be obvious, EPI put a fair amount of effort into setting up these assays, both the PCR and also the serology. They were taken advantage of in a number of different settings. This just sort of begins to run through some of the areas where we were involved in community-based testing. From medical and lab staff here at UF, we ran a total of 264 samples. We were involved with a major testing program at the villages. Mike Lauzardo played a critical role in setting all of this up, and for us it was sort of a good testing ground to work through rapid community-based testing programs. Over a period of weeks, we ran over 2,282 to be exact samples for residents of the villages. Top picture shows out on the polo grounds in the villages where we set up tents and set up operation. We were engaged in studies in association with UF Jacksonville, looking particularly at some of the minority communities that are within that neighborhood that are linked with some of their programs. Close to 700 samples there. First responders, I'll talk about that a little bit more in a minute, over 3,000 samples. Next slide. We did an island. We went out and did Cedar Key at the request of Cedar Key, over 500 samples there. Top picture is the group that went out to do the Cedar Key group, the Cedar Key testing. We've been involved with the folks who go to provide medical care at Grace Marketplace in San Francis House, close to 400 samples there, looking specifically at homeless populations within Gainesville. We've been looking at agricultural workers, have a major program underway at PK Young. You can see the folks in the middle picture there, middle lower picture, lined up in the driveway waiting for folks at PK Young. And again there, Eric Nelson, Sarah McCune, and Tony Morelli have played a key role in pulling that particular research project underway. And we've been involved with some of the folks in GI here, done over 700 samples on endoscopy patients. Picked up one that we didn't know about in advance, but it's been interesting seeing how we can apply some of these techniques to the screening of clinical populations. In many of these instances, we also collected serum for serology. And in all of these instances, people from whom we collected samples, we got signed consent forms and actually have the ability to go back and to obtain further samples and further data to monitor changes across time. So this has been it's been quite a trip to go through, to set up these community-based studies, and to use them as hopefully as a basis for better understanding the long-term natural history of coronavirus, of COVID, SARS-CoV-2 within this region. Next slide. We've really put a lot of effort on the first responders study, and again, I particularly credit Lisa Merck, who's played a key role in that. This is at a point we had 1,677 participants. Actually, I think these data are fairly up to date. You can see the types of populations that we have tapped into. Again, we started this because even though testing was becoming available for patients, there were initially prohibitions against screening anybody if they were not symptomatic. And so we had major concerns about first responders, frontline individuals, people that were involved in frontline work with the public. And by using the research protocol format and taking advantage of the facilities, the capabilities within our rapid PCR lab and the serology labs here at EPI, we were able to provide these services. Next slide. And again, this just gives some of the data. You can see that over on the left is the number of samples collected weekly versus the percent PCR positivity. At the beginning, we were doing a lot of samples. We're doing fewer now, but we are starting to see a little higher positivity rates. And again, over on the right is some of the serology results. Again, a lot of the initial testing when we were first getting things underway. But now that we are beginning to follow individuals who were indeed infected, you can see that the serology positivity rates are going up. Next slide. So at this point, what I'd like to do is let some of the other folks talk about some of the projects they have underway. Joe Basisi and folks over on environmental and global health have been involved in a series of studies of wastewater analysis. Joe, do you want to talk a little bit about what you've been doing?
Sure. Thanks, Glenn. And thanks for the opportunity to talk a little bit about some of our work that we've got going. I was part of that group that set up the rapid PCR lab back in the beginning of the pandemic, and but my background is actually more in environmental toxicology and environmental sampling, and so it made sense to transition to doing wastewater analysis when that became apparent as a method for trying to track transmission of the virus, both here on campus as well as in cities across the U.S. So back in about April, May, we started to develop our method here at EPI for extracting the virus from wastewater. Started collecting samples in Cedar Key and the city of Gainesville here in our 2 wastewater treatment plants, and we've been tracking those the whole entire time since May and June of last year. We've had some really nice agreement between. And then as we started bringing folks back to go to classes in the fall, we decided to try to monitor this on campus to really try to assist the Screen Test and Protect team with their efforts to decrease transmission on campus. So in working with UF facilities and UF housing, we were able to identify a number of different residences here on campus that include fraternities, sororities, dormitories, graduate and family housing. We cover about 85% of the campus residents here on campus. And we were able to get about 28 different sites that we have been sampling on a weekly basis since back in the fall, the beginning of the fall semester. Continuing that right now. So every week our team goes out and samples those 28 different sites. We have rapid turnaround extraction from the wastewater, and then those results get reported directly to the Screen Test and Protect team, and that really helps them to do some targeted testing messages, try to get folks to be more aware and potentially get tested if they're in a building where we may have high levels of the virus being detected in the wastewater. So we've had some really interesting results, like I said, here as well as here on campus. We've got some really nice confirmations of residence halls where we know we have high numbers of cases corresponding well with levels in the wastewater, as well as measurements in wastewater that don't necessarily pop right up with numbers of cases. So it's been a really interesting study. It's been tremendous to work with the Screen Test to Protect team to help this campus effort. So thank you very much.
Okay, Joe, thanks. I'm going to ask Mike Norris to talk a little bit about some of the work that he's been doing, more laboratory-based, but again, sort of giving a sense of the range of activities that we've got underway here. Mike?
Thank you, Dr. Morris. I'll just tell you a little bit about 4 collaborations we've had going since April. They've all sort of coalesced around this idea of identifying inhibitors of SARS-CoV-2 replication so that we can develop novel therapeutics. The first collaboration is with Dr. Stephanie Karst and Dr. Christopher Volpe, where we use CRISPR knockout and CRISPR activation technologies to screen for host factors that are required for SARS-CoV-2 infections, and also to identify host factors that restrict SARS-CoV-2 infections. We have a lot of data, we've done a lot of these screens, we obtain these sort of genetic networks on the left side of the slide here, and we use those genetic networks to identify drug inhibitors. And UF Innovate asked me not to share a lot of the information I originally had on the slide because they're protecting that intellectual property now. So suffice it to say, we have 6 drugs from these screens that show in vitro inhibition of SARS-CoV-2 induced cytotoxicity. It also inhibits their replication in those cells by real-time PCR measurements. Some of those drugs are at various levels of FDA approval for other purposes. We'll continue our research on that. Another collaboration that we're working with is Dr. David Ostrov and Dr. Leah Resnikoff. And we had an inkling that antihistamines may inhibit binding of SARS-CoV-2 to the surface of cells. We published this in Biochemical and Biophysical Research Communication Journal a couple months ago. We found a couple over-the-counter and prescription antihistamines were effective at inhibiting SARS coronavirus 2 replication in cells. We also found in that paper that we looked at a cohort of 200,000 patients in California. We found that if they had taken some of these antihistamines prior to getting a COVID test, they were about 2 times less likely to test positive. So it was an association study, but it still was something to go on there. And our next step there was to work with Dr. David Ostrov again. And we asked, well, are there other over-the-counter compounds that can synergize with diphenhydramine, which, if you're not familiar, is Benadryl? Can we find a compound that synergizes with diphenhydramine to inhibit SARS coronavirus 2 replication? And again, drug A, when mixed with diphenhydramine, significantly inhibits its replication in cells and reduces the cellular cytotoxicity associated with infection. And right now we're working with some industry partners to develop a COVID Theraflu. That's not the trademark name yet, but we're working on it. And then in the last collaboration, again, we're working with David Ostrov and Dr. Chris McCurdy in College Pharmacy. He provided these highly specific receptor ligands, so different receptor than ACE2, but again, these receptors are very well characterized in the ligands are very well characterized in their binding to these receptors. And we identified 3 more drugs that showed the ability to reduce SARS-CoV replication in vitro and reduce cytopathic effects in cells. So all these basically, the next steps will be testing in the in vivo mouse model of COVID infection. And we're on track to setting up that ABSL3 lab mid-March. We're working with ACS and EHS to get that going. Thank you.
Right, thank you. Next, if I can have the next slide. We have here at UF, EPI, probably the premier aerosol research group, particularly looking at culturing of viruses such as coronaviruses and others, respiratory viruses here. And that's resulted in some outstanding publications which have generated fairly impressive altmetric scores. So if John Lednecki is the key in this, and so John, do you want to talk a little bit about some of the stuff you've been doing? John, you on?
Thank you for giving me this opportunity. What you're looking at here in the middle is a commercial version of an air sampler that was developed jointly by a commercial company and UF engineers. We had a real big hand in improving the way this instrument can collect virus particles from the air. And it turns out collecting virus particles from the air is very difficult, very technically difficult. And collectors that are commonly used typically destroy the virus particles they collect. And if you do that, you can't do a risk assessment. And by that I mean the air you breathe is absolutely full of virus particles, bacterial viruses, plant viruses, and so on. And we only worry about the viruses that are viable. That's a correct way of saying live, that might cause infections in humans. Using this instrument, which is better than pretty much anything you can buy on the market otherwise, we did a pilot study in the Student Healthcare Center, and we were able to isolate adenoviruses, different coronaviruses, influenza viruses, and so forth and so on. And we isolate these viruses in cell culture. So not only do we identify what's in the air, we grow them, showing that they're infectious. And what's remarkable about this particular study we did was this was at the time when no one was wearing face masks, and it was assumed that the air in the Student Healthcare Center really wasn't very dangerous, posed no risk. And you see, we saw all these viruses that we found during influenza season. Can I have the next slide, please? Now, using this equipment and a similar approach, we asked whether we could find viable SARS coronavirus 2 in a hospital room that had patients. Now, a lot of people had tried this and failed. And we chose a hospital room thinking that other viruses wouldn't, that might be present in air wouldn't be present and confound the results. Because if you try this type of work, other viruses can outgrow the virus you're looking for. And we just so happened to have a situation where one patient was being discharged. The patient had neurologic COVID and wasn't producing the virus, and a newly admitted patient, so sort of at the acute phase. And when we did our air sampling, we were able to show that viable virus could be collected at long distances away from the patient. And we were able to link the virus we found in the air sample with the virus that was in the patient who had acute respiratory COVID. So this was the first time this was actually ever successfully done. And to my knowledge, not too many people have really been able to succeed. And I attribute that to having this air sampler that was co-developed here at UF, along with testing at the right stage of infection. We've done a similar study using a different air sampler in a car driven by someone with mild disease. And we've been able to show that once again, infectious virus particles are present in the air. That tells us that there is a risk posed by riding in a car with somebody who's got an active case of respiratory COVID. and the virus particles, by the way, are in the inhalable range mostly. That means that the size of most of the infectious virus particles is relatively small. They're in aerosols that can be inhaled into your lower respiratory tract. Next slide, please. One of the last things we've done is we've been looking at the viruses that are in circulation. So as Dr. Morris said earlier, we had been isolating virus from patients at Shands. Most recently, there was a question whether one of these faster spreading variants of SARS coronavirus is in the area. And we happen to have a patient that for which preliminary studies suggested that one of these faster growing variants was found. And indeed, we did direct sequencing on sample from that patient. And what we found is the virus has distinct mutations. It's definitely within the clade, like the faster spreading ones from England. So that was the progenitor. The interesting thing about the virus we found is it's similar to viruses that are spreading in Florida in that it has a unique mutation, and that mutation sets it apart. So the one from England is called B117, and now there are two clades that have derived from that. One is the California clade, and one's the Florida clade. The Florida clade, again, has the unique mutation, but what's interesting about it is it seems to be spreading much more quickly than the California version. And we don't know if that has to do with the unique mutation that the Florida clade has. And that mutation is in its RNA polymerase. So this is something that remains to be explored. And I'll leave it at that.
Okay. And to sort of kick this up to a level with a greater number of samples. Marco, you want to talk about some of the stuff that you're starting to work on in terms of the more statewide type activities?
Right. Yes. Thank you, Glenn, and thanks, John. I mean, that was the perfect preamble for what we are trying to do next. Of course, there has been a lot of discussion, and especially in the past few months, about all these new lineages emerging. There is mounting evidence that some of them, like the B117, that is the UK lineage, might be more transmissible. I don't think there is a clear answer yet, as John was saying, whether or not, in fact, these lineages might be inherently more transmissible or more pathogenic, but certainly we seem to be heading in that direction. And there is an urgent need to assess the prevalence and the incidence of lineages circulating, especially here in Florida, like everywhere else in the world, but on large numbers, of course. Just to give you an idea, we have about 1.8 million people infected in Florida, as most of you know. But since January last year, so in the past 30 months of the epidemic, basically only 3,500 sequences have been obtained statewide. So there is really a need for scaling up, especially now that the vaccine is available. And there is, of course, the question of whether or not also vaccine-resistant strains might emerge into the future. So we have been working very hard together with Glenn, of course, and Mike Lauzardo, and people at ICBR, people at Path Labs, and Kenneth Rand and other collaborators at Shands Hospital, to put together a genomic epidemiology consortium that will have the job to do genomic tracking and genomic monitoring of coronavirus diversity, and not just here in Alachua County. I've been involving people from the University of Central Florida, specifically Taja Zarian, and people from the University of Miami, among others, Mario Stevenson, who is the director of the CFAR, the Center for AIDS Research, who has also been involved actively monitoring the coronavirus epidemic. And so the basic idea is to start obtaining and storing things that in the next slide that have been already started, start collecting here at EPI a large number of samples from all these different sources to characterize them, sequence and characterize them, and with phylogenetic analysis and other tools that, of course, my lab has been working on for many years. So this is a schematic representation of the sequencing pipeline. Right now, Path Labs in town, in Alacho at least, and this doesn't take into account the samples that potentially might come from Miami and from the University of Central Florida, is testing around 5,000 samples per week with an average of 300 to 600 positive cases every week. Now, this number hopefully will decrease in the next few months, but depending on the emergence of new strains or vaccine-resistant strain might fluctuate. So the basic pipeline would be to be acquiring. We have already started. We have already collected in the past few weeks nearly 2,000 samples that are currently stored at EPI. So for with the pipeline and workflow that we put together with ICBR, starting from 300 to 600 samples a week, we can do RNA extraction using the robot, the Hamilton robot, in 2 days. Run NGS. I said here 14 days, but this is actually an overestimate. We can probably do 600 samples easily in 1 week, maybe even less, the moment we optimize the pipeline. And again, another 7 to 10 days for the in-depth bioinformatic analysis, phylodynamic analysis that essentially are going to look at transmission clusters, estimate basic reproductive number for each one of the clusters, look for new lineages, and use artificial intelligence algorithms to predict epidemic spreads. Overall, we basically we are putting together a system that will be able, when we will be up and running, to generate essentially about 2,000 new sequences from different sources every 4 weeks. And this will happen continuously, so there will be overlaps. And we are in touch with Department of Health officials. And the idea is that eventually we will be able to basically have reports, weekly reports that will be sent to SHANS, public health officials, and so forth about the status of the genomic epidemiology of COVID. And if I can have the last slide. This is not just, of course, potentially, of course, a gold mine for research, but in fact, the findings and the questions that we are going to ask have immediate relevance, both from a public health standpoint and from a clinical standpoint. Specifically, I will just go through these 6 points. We are going to detect and quantify circulating viral lineages, including the ones potentially that are more transmissible, more pathogenic, or novel lineages like the novel Florida lineage that might be already circulating, as John was saying before. Detect and characterize transmission cluster, assess contribution of asymptomatic and children to viral dissemination, which is still a big open question. Assess the effect of vaccination on transmission clusters and detect potential emergence of vaccine-resistant strains. Develop artificial intelligence-based models of viral spread and forecast future outbreaks in at-risk population and areas. And finally, we will essentially develop an integrated sequence, a clinical demographic database, and a genomic dashboard that will be updated regularly. And we'll work a little bit like the Johns Hopkins website, but focused on genomic data. We are starting our sequencing effort. We have about 2,000 samples already, using some money that we have through endowed chairs and some previous research grants, basically within the next 1 or 2 weeks. So we hope to have the first 2,000 samples sequenced within the next 4 to 6 weeks. And in the meantime, I'm putting together, a pretty interdisciplinary team to apply for a large NIH grant that hopefully will give us sufficient funds to keep doing this, not just to sequence the samples that we already have, of course, but in fact to keep collecting and keep monitoring the situation, at least for the next year and certainly for the next few years. And that's all I have.
Okay, thank you. Shift gears a little bit. We also have one of the strongest mathematical modeling groups in the country here, headed by Ira Longini. I would note, for those of you who may not have noticed, on our EPI webpage, we have a tab specifically for math models. As the pandemic has progressed, there have been ongoing questions about models, and so to facilitate the availability of models for both public health officials, but also for investigators within the state, we have been maintaining and publishing models in a moderately regular basis. I just noticed it's been September since we put other models up there, and we need to update some of the model status. But Ira, you want to talk a little bit about your program?
Yeah, thanks, Glenn. It's a great pleasure to talk to this group, our group. And so, as Glenn mentioned, we're doing a lot of modeling, and our immediate group here at UF, I've listed it as Tom Laddish in biology, Natalie Dean in biostatistics, and Yang Young in biostatistics, plus a group of our PhD students and postdocs. Next slide, please. And we do have also a center which is spread. We have a local center here, C-Squid, that's at the EPI and Department of Biostatistics. But we also have a larger group. So I've listed that group as well, and doing all this modeling work. So there's a group at Northeastern University headed by Alex Vespignani, at the Fred Hutch by Elizabeth Holleran, and then some other centers as well. We work together as a network on doing the modeling. Next slide, please. And in addition to that, we're deeply embedded, especially me, within the World Health Organization blueprint to prevent epidemics. And you may say, well, we're not doing a very good job in this effort at WHO, but it's really to prevent and/or respond to emerging pathogens. And certainly coronaviruses were on our radar screen for the last 5 years. And so we're also involved in that effort, WHO. Next slide, please. So rather than show you, examples of all the work we've done in the few minutes more I have, I just wanted to summarize the research areas. So these are our current research areas, but also where we're going in the future. And the first is we're very involved in design, analysis, and conduct of interpretation of COVID-19 therapeutic and vaccine trials with the WHO, and that includes a large Solidarity vaccine trial, which is a multi-vaccine, multi-country phase 3 platform that's going into the field almost as I speak now. We're also work on the design, analysis, conduct, interpretation of COVID-19 vaccine effectiveness studies as vaccines are rolled out. So those are large collections of studies, everything from test negative designs to cluster randomized designs to try to estimate the effectiveness and impact of vaccines as they're rolled out. And then we're also working on the mathematical modeling of COVID-19 transmission and control with both non-pharmaceutical interventions and now vaccines on local, country, and global levels. And we've done quite a bit of modeling, which I guess we need to put up on the EPI website for the state of Florida, projecting what to expect over the next year with different assumptions about vaccine rollout. Another project is we're developing mathematical models and observational studies to come up with optimal use of COVID-19 vaccines. As you know, the current strategies are way too slow and inadequate, with these very slow rollouts starting with high-risk individuals. and then moving, down the age group, well, first responders and so on, then slowly rolling out month by month to other age groups and other segments of population. And that's, going to be very slow and ineffective in Florida as well as the U.S., as well as many other countries in the world. It's projected most countries in the world, especially developing countries, will get enough vaccine at most to cover 20% of their population. So we're developing methods for to try to speed up the control with different types of targeted, even ring vaccination, to try to speed up the control on all these different levels. And then our last activity is we do quite a bit with the statistical analysis of COVID-19 transmission studies, pathogenicity, severity parameters from epidemiologic studies, such as estimating transmissibility in household studies, say, of children and so on. So next slide, please. I thought I would just mention, next slide. Let's see, I had another slide that seems to be gone, which listed some of our publications. So I won't talk about those. That was how I was going to summarize what we've done over the last year. But so I'll stop there. Thank you. Maybe you go back a slide. Maybe I missed that slide. Go back. Nope. I guess that one was gone. I must have hit the slide limit. So thank you.
And I'd like to thank Ira. And I would like to note in particular, I think most of you are aware we had a COVID-19 vaccine seminar series, which included talks by Ira, Natalie Dean, and Tom Laddish from the group here at UF. These are, I believe, still posted on our website. The lectures are outstanding. And I would strongly, if you're interested in these topic areas, I would strongly encourage you to go back and take a look at some of the talks that were given. Next slide. And actually, at this point, I'm going to shift over and ask Derek Cummings to talk about some of his activities. And again, Derek, I'm not sure, we may have your slides out of order, but go ahead. I'll hand this over to you.
No, that's the right order. I shifted those around. So,
okay.
Yeah, so yeah, I just wanted to briefly describe some of the work that that our group, the Infectious Disease Dynamics group at the Emerging Pathogens Institute and the Department of Biology have been doing, and we've been doing a mix of modeling work, theoretical work, field work, and some laboratory work with a mix of collaborators, many here at UF, but other institutions as well. And so, just to mention briefly a number of the projects that we're working on. So working with Maureen Long, Lisa Merck, Glenn, Cindy Montero, Eric Nelson, and others here at UF, we've been characterizing serological immune responses to SARS-CoV-2 and also a specific interest of mine, mucosal immune responses and sort of upper airways. And so we're utilizing samples collected here at UF and working with collaborators at the University of Manitoba to develop an assay to look at mucosal samples, anterior nasal swabs, to see if we can detect prior exposure to SARS-CoV-2, and also see if this might be part of the story of why there is such heterogeneity in clinical presentation, that there might be immune responses that are really limited to the upper airway and not systemic, and that might contribute to the variation that we might see in serological responses. Where that's ongoing work, we're also starting a vaccine effectiveness study in collaboration with the Yale Health System. So we are using both passively and actively collected data from a 1 million person plus health system data set to measure aspects of the currently utilized vaccines and anticipating future vaccines, to measure aspects of their effectiveness and look at durability of protection and also look at 1 dose versus 2 dose and a few other characteristics. We are about halfway through a 5,000 person serological survey of people living in Chennai, India, a city of over 10 million people. We're doing a random representative sample of households across the city to look at serological seroprevalence in that city, which has had sizable clinical burdens, but really not a lot of characterization of how widespread the pandemic has been in Chennai. And we're also looking at within city variation to see within this large metropolitan area whether we're going to see strong stratification both spatially and along a number of different demographic characteristics of attack rates. Similarly, we've analyzed a large serological household study in Geneva with collaborators from Johns Hopkins and Doctors Without Borders. And we've also been doing quite a bit of modeling to try and understand the impact of non-pharmaceutical interventions, and this is a collaboration with Sandia National Labs and Johns Hopkins and others. That has utilized mobility data derived from cell phone based geolocation. And the last thing I'll mention in a little bit more detail is trying to interpret cases, deaths, and serological data simultaneously to estimate infection fatality rates and cumulative incidence in multiple parts of the globe. So next slide. So this past fall, we published in Nature an estimate of infection fatality rates across 42 different countries, including 25 that had serological data to go along with their case and death data. And as has been described in a number of different locations, describe this strong age-specific trend in infection fatality rates, where those over 80 are having infection fatality rates around 10%, whereas those in younger age groups are having much lower infection fatality rates. It does break out by men and women quite strongly, but it is surprising. The overall trend is that there's quite a bit of similarity across contexts, particularly when you stratify data to look at, let's say, under 65s, where reporting for those over 65 might be very variable. And next slide. One of the ideas that we've done is if this infection fatality rate has been fairly conserved in different locations and by time, can we use deaths to reconstruct how many infections have occurred in different parts of the United States or in different parts of the globe? And so this is in collaboration with the US CDC. We've been integrating case, death, and serological data to estimate cumulative incidence of SARS-CoV-2 in the US over time. And so these maps are just showing the percentage of the population that we estimate have been infected over time. And we are working to incorporate the waning of immune response and detectability into these estimates, and just look at the consistencies of these different data streams to try and estimate the burden in different locations, and then the present status of immunity and seroprevalence in different regions of the US. And that's my last slide.
Eric, thank you very much. And again, we're going to, at this point, shift gears a little bit, and I've asked Mike Lauzardo to provide a little more to provide some details on what's been going on in terms of the overall pandemic response on campus and community and within the community, talking specifically about some of the role that's been played by EPI. Mike?
Great. Good afternoon, everybody. It's a pleasure to be here with you guys. So can you go ahead and get the next slide? Thanks, Nicole. So just kind of as a review, what I wanted to go over here today was to talk about what we're doing on campus, but obviously in the context of research and the role of EPI. Also talk about our activities to kind of say some of the data that we've collected, not necessarily with the purpose of doing research, but more operational research, and then also as an opportunity for us to tap into this data with some important caveats in there as well. But let me just kind of review things briefly. Many of you have had to deal with some of the consequences of our policies on campus, and just a little bit more in the background for those of you who may not recall exactly. But our strategy to control COVID on campus was essentially to create a freestanding public health surveillance unit. It's a long story, but through our collaborations with the Department of Health over many years and other areas, really those ties really were critical for us to be able to do the activities that are necessary to control COVID in terms of contact tracing, isolation, quarantining, having that authority, and being able to implement some of the public health procedures that needed to be in place to control things. We did this during the fall semester, and now that we've been able to increase our capacity with testing, and I'll talk about that briefly later, as well as our other activities and collaborations that you've heard here already, primarily the wastewater epidemiology program and some of the other things that you've heard here earlier. Those are going to be important steps to help us move forward. I almost tried to click on the slide here, forgetting that you have control, Nicole. So if you can go to the next slide, please. So again, the whole idea of screen, test, and protect for many of the students, maybe some of your graduate students, what they've really felt has been the testing requirements that have been included in there. Many of you get these emails that come back and forth and may have actually had to be part of the targeted testing. But what we really sought to do is to create a comprehensive approach. No single way was going to get us out of it. So in other words, it's not just about testing, not just about contact tracing, but a lot of details that go into it. So we really looked for the most comprehensive approach to doing that. So we have surveillance testing this semester increased by having mandatory testing amongst those groups that had the highest test positivity rate, were therefore the highest risk last semester, applying that knowledge to this semester. You get the weekly symptom screening questionnaire, which I originally, when we started doing that, I was somewhat skeptical, but we've actually found that we've actually picked up a large number of cases by using that simple questionnaire and reminding people. So we've actually had a pretty significant amount of data that we've collected related to that. What I call targeted testing, so in other words, doing the testing around identified contacts or identified cases, primarily in congregate settings, so really the dorms and the Greek life residential facilities. You heard about wastewater epidemiology and been so thankful for the opportunity to work with Joe and Tony and the rest of the team. That's been extremely helpful, and now we're really gaining some of the benefits from that. The one point that I want to talk about is that the disease investigation program that we've got, many of you have come into EPI, you see that the conference rooms are taken up by a lot of young folks on computers. Those are our graduate students, and a lot of them are many of your students as well, epidemiologists, both who've graduated and also folks in the graduate programs. They do more than just contact tracing. They do a detailed 30 to 45-minute interview to get a lot of detail to look at exposure risks, and all that data goes into REDCap. REDCap manages so much of what we do, including the testing, the release from campus, all of our surveillance data, all of our exposure data. All that is in REDCap and actually in a very usable form longer term. I'll talk a little bit more about that later. And then we've got our operational surveillance studies that we do on campus. So we're trying to hit everything at one time as we continue to try our best to kind of control COVID on campus. Next slide, please. This is our epidemic curve taken from our website on the Screen, Test, and Protect website. It's don't have this month's epidemic curve. I haven't updated this slide, but you can see the peaks and valleys. But again, the reason why I put this here is that this kind of belies a lot of the data that exists in REDCap related to that. So we've got a lot of data with regard to testing, individuals who've gotten tested, the contacts, what happened with those contacts, and a lot of the sort of surrounding information related to them, as well as their demographics. Next slide, please. And this just shows the ability that we've had by working with our partners at UF-Path. They've been fantastic. They've really scaled up, and this is actually right before the Thanksgiving break that shows the scale up where our positivity rate was kind of drifting down slowly. In terms of scale, it makes it a little bit difficult to appreciate. And this, at this point, was the highest number of tests that we had done in one day, which was 2,200. Now we regularly get close to 3,000 tests per day. And again, that's a testament to a lot of the work on the team, as well as the folks at Path Labs who've been fantastic. An important thing to remember is that these specimens are all linked back to the demographic data that we have on each patient, including all the positives, as well as their isolates. So again, we've been disappointed with some of the data repository issues, and that's some challenges that I think added gray hairs to both me and Marco and the others that have been trying to get this moving along. but still the data is there, and there are a lot of opportunities to do some good science moving forward, especially as we move forward on understanding the variants and the impacts that those have. Next slide, please. And here's some of the data that just in terms of just raw data in terms of our numbers with testing. We created a lot of different categories, and these aren't all of them, but of different kinds of students to look at relative risk. This kind of talks about the relative number of percent of tests that we've done amongst the cohorts that you see on the left. But really beyond this, we've got details with regard to their percent positivity and their association with cases. We also have maps of where cases occurred on campus and detailed analyses of each one of the cases. So in other words, we've got a very good look at kind of what that what the pandemic has looked like on campus in terms of the data and what that has looked like overall. Next slide. And again, I think this one just shows the percent positivity, and again, showing the relative risk with looking at, say, for example, students in Greek housing and those who are not in housing, those in residence halls, and then comparing that to faculty and the positivity rate amongst those who've been tested. In addition, we also cross-match with the state health department. So a lot of this positivity is not just with our testing, but that's those who have ever tested positive anywhere in the state at any time. So again, we've got a very robust series of data and opportunities to look deeper into the data. Next slide. In terms of what's going on now, it's kind of an exciting time because we've been able to scale up and operationalize a lot more testing. These peculiar boxes are little warming chambers that we built for our poor testers who are out there. Yes, it is Florida, but 30 degrees in the morning is cold anywhere where you are. So we actually have little space heaters in there and have been able to operationalize large scale testing. We actually have now been able to do, and we're trying to avoid doing this, but on our first day of doing vaccinations on a campus site for the community, we did 1,800 tests and 200 vaccinations at the same time at one location. So again, a lot of logistics have gone behind that, and again, a lot of operational considerations. You've heard about the wastewater surveillance. Marcos talks about the sequencing project. The other things that we've just been identified or actually selected as a vaccine clinical trial that's going to be looking at the impact of vaccines on cutting down on asymptomatic infection amongst the college student population. So we should start recruiting within a couple of weeks. And that goal there will be, I think our target is to recruit 1,000 students, which I don't think will be much problem given the size of campus and the interest so far. Next slide, please. And here's the really exciting part, just to talk about our community efforts and kind of what we've been doing. Again, I mentioned about the health department partnership, but I think Eric Nelson's on the call, but Eric and others on this call, I'm sure, have been part of the Science and Medical Advisory Committee for the Alachua County Health Department as we work with the public school systems here in Alachua County. We've also been doing a lot of significant vaccine scale-up. Again, no offense to Ira and the others of you who have been involved with vaccines. Vaccines are useless, right? It's vaccination which matters. So vaccination, getting that last mile, getting the vaccine into people's arms is that critical step. And to doing that, we've got a number of different initiatives going on in the community now that would give a lot of opportunities for not basic science or other sort of the research that we're usually associated with at EPI, but also to bring the social scientists to kind of look at the best ways to reach out to vaccine hesitant populations. We've got great relationships on the east side of town. The photos that you see on the right include our drive-up vaccine clinic that we did a couple of weeks ago. We're doing one right now where we're vaccinating 600 people as we speak through the community on a drive-through at the same location. And the picture at the bottom is at Mount Moriah Missionary Baptist Church on the east side of town, where we did 364 community members on the east side of town in a great collaboration that's continuing to grow through our relationships there. To come, we're looking for farm workers and kind of reaching out. And again, with each one of these populations, there's great opportunities for research. We're focused on operationalizing this and for implementing the science that we've got available. But really, there are a lot of opportunities to have linkages to other investigators. Outside of kind of our STP role, but certainly within our bailiwick in terms of COVID and our relationship with the community, we've got a monoclonal antibody rural access project going on where we've developed an early warning system. That project is rolling out in Columbia County and in surrounding rural counties here shortly. That's an HHS funded and supported project that's being run through CTSI that we're a part of. And also to mention the community education and outreach. And again, the list could go long, but in the interest of time, I just wanted to go through it quickly. But working with the Gainesville Pastors Association, the Black on Black Crime Task Force, Rotary, and the Vaccine Access Steering Committee, which we formed a couple weeks ago and are going to have our second meeting this Friday. So all these really create, next slide, please. A lot of opportunities for research data that we've got collected, and also relationships that we've got that will help foster these type of investigations and other opportunities for the folks here at EPI and others on campus to engage with the community in a meaningful way. We've still got a lot of heavy lifting to go, a lot in terms of testing and vaccines over the next couple of months, certainly, plus a big push for vaccines later, but really a lot of exciting opportunities that we can help facilitate research and in fact play a role with many of the different aspects that have been discussed here earlier. So thanks for your time, and appreciate if you have any questions, just let me know.
Okay, Mike, thank you very much. Again, given the way the time is going, I think what I would ask is if people have specific questions or want to do, link up with individual speakers, if you could use the question and answer boxes. It looks like there have been some good conversations going on within those. And so I would encourage you to continue to take advantage of that, and obviously talk to folks outside of this particular setting. Again, the whole point of this today was to try to facilitate communication among investigators, particularly at a time when labs are locked down and folks aren't on campus, so that people have an idea of what's going on. And hopefully we can build further bridges among what are some of really, outstanding investigative teams, many with national, international reputations, and see how we can work together to try to better deal with the current pandemic. What I'd like to do at this point, that's all the formal presentations that we have. What I'd like to do at this point is to shift a little bit and talk a little bit more specifically about EPI and where we are, sort of as a prelude to thinking about, where we're heading, and kind of given the fact that we're now hopefully beginning to move outside of the pandemic curtain, and to begin to look at, ongoing questions, opportunities, and directions that we might be taking as an institute. So, I've got a couple of slides here that just sort of summarize who we are as an institute. We have 212 members at the faculty level. 5 of those are outside the UF domain. We are happy to take members who are not UF faculty. We have 473 associate members, which are basically postdocs, grad students, undergraduates. And we've got 236 what we call EPI friends, who are probably about half of those are outside of the UF domain, essentially people who are interested in the type of activities in which we are engaged and have been partners with us frequently in the community in terms of the activities we have underway there. Next slide. Next slide. In terms of the actual physical facilities, we've got 297 people who are assigned space in the main building, and another 52 collaborators have a building access. This includes 51 faculty and 246 student interns, graduate students, postdocs, and staff. So a lot going on within the building, but if you'll compare to the previous numbers, there's a lot going on outside the building as well. And I think what I would emphasize is that part of the role of the Institute is to try to facilitate the communication across campus, across multiple colleges. Our BSL-2 labs are full, our BSL-3 labs. We currently have 5 of the 16 BSL-3 labs that are committed to work with SARS-CoV-2. I will say one of the problems we have encountered is that there are a number of individuals on campus, investigators who want to do work with SARS-CoV-2. We have the laboratory space. The problem is getting people who are BSL-3 certified and who are able to work within the BSL-3. And so we're trying to get through that bottleneck, but at least at this point in time, we continue to have an urgent need for people who are BSL-3 certified and who may be interested in getting involved in specific projects with SARS-CoV-2 at the BSL-3 level. Next slide. But I would note that we're not just here in Gainesville. I would say, unfortunately, we weren't able to get some slides from Roel. He has a significant international presence, but to mention some of the others, we do have a strong presence in Haiti. Over on the left of this slide is a floor plan of the laboratories that we have recently, where we've recently finalized renovation that are associated with the Lovechild compound in Font-Parisien, Haiti, adjacent to the very large clinic that they have, as well as a large birthing center. You can't really see on the slide what all is here, but we actually have an insectary inside the building where we can do studies with mosquitoes and with arboviruses. Picture of the main lab area. This is a team, including some of the folks from up here at UF who are engaged with our activities in Haiti. And in Haiti, as in most other places, it's not just bench work. There's also digging the car out of a hole when we get stuck. So for those of you who have an interest in engagement in international activities, particularly in Haiti, you know, we clearly have the laboratory capabilities there, and I would strongly encourage you to think about expanding projects into areas such as Haiti. Next slide. We also, besides the laboratory at Font-Parisien, we also work closely with the laboratory closely with the School of Medicine and Pharmacy of the Haitian National University in Port-au-Prince. This was a conference we had year before last on water issues. These are the folks from UF which were attending, but we have actually an assigned laboratory space in the School of Medicine in Port-au-Prince. Because of travel restrictions, we have not been able to fully utilize that space, but again, there are substantial opportunities there. Another area where we have recently opened up work is in Democratic Republic of the Congo, DRC, and specifically in Goma. Actually, I misspelled that. That's not Coma, it's Goma, G, in North Kivu. These are the laboratories with which we are collaborating. Amy Labo Lab, which is the provincial health laboratories. There's an inside view. We actually have a whole container load of equipment which is due to arrive there in the next few weeks. There clearly is a need for, you know, upscaling, upgrading, and increasing capacity. But we're already in discussions not only with studies with cholera and diarrheal disease, but also becoming engaged with some ongoing COVID studies that are taking place in Goma. Goma's got a population of 1 million people. It's in the Rift Valley, right on the lake, with a active volcano towering over the city. It's an amazing place. So again, the international opportunities are there, and we clearly have a footprint in a number of different areas. For those who are interested in these areas, I would encourage you to talk with me or others who are engaged with some of the international work. Next slide. This is a slide that shows the total extramural awards for all EPI members over the last several years. You can see that up through FY20, the numbers of awards were increasing. For FY21, that's year to date, which is one reason why the numbers are lower. Hopefully they will come back up. But I will also say, I think many of us are recognizing there have been difficulties with funding, both in maintaining older grants because of restrictions imposed by COVID, but also in getting new funding. I know, you know, it has, even with outstanding scores, grants sometimes are not getting funded in NIH. So I think as we look forward, there needs to be an awareness that the funding climate is at this point, I'm not sure the right word, but maybe unstable or not as stable as it has been. And so that's one reason why, as an institute, I think we need to be working together to facilitate ways that we can build on the expertise that's available locally within the institute and across the campus to try to maximize our success rate in terms of extramural awards. Next slide. And again, this is the sources of extramural funds. This pie chart has stayed remarkably stable over the last decade. We basically run, in this case, 44% NIH funding, but it runs anywhere from 44 to up to 50% of NIH funding, with then a wide distribution of other sources, Department of Defense, CDC, other feds, USDA, foundations and nonprofits, NSF. And so again, I think as we move forward, as we begin to move, to a situation where COVID is no longer the dominant emerging pathogen, we need to be thinking about funding sources and ways in which we can utilize the expertise that we have within the Institute. Next slide. Publications, again, over the last couple of years, we have seen a steady increase in publication number, which is very satisfying. Again, year to date for FY21 is still low. I'm hopeful that those numbers will go up dramatically as we get further into the year, and hopefully as some of the publications related to SARS-CoV-2 start coming out. Next slide. Research Day has always been one of the major times when we've been able to bring together the group that constitutes EPI. This is a slide, pictures from last year's Research Day. Next slide. This just shows topic areas presented last year. You can see vector-borne diseases at that point in time constituted 31% of the abstracts, which sort of gives a feel for where a lot of our activity has been focused. You can see a variety of other topic areas out of the 143 abstracts submitted. Next slide. And again, I would emphasize the fact that this is clearly a multidisciplinary institute. The largest contribution in terms of abstracts was from IFAS, but strong representation from Vet Med, from class, from PHHP, medicine, so and as well as a fair number of abstracts externally. So overall, I think I'm pleased with where we are, but I think we probably need to begin to think about where we want to go, taking advantage both of the expertise that we have within the COVID field, but also looking for, moving forward, what we need to be doing as an institute. And actually at this point, we do have another research day coming up. Needless to say, this will be a virtual research day. And I've asked Dwayne and some of our IT folks just to provide a very brief introduction to exactly what our research day is going to look like this year. And then after we do that, we'll see if maybe we can take a couple of questions or maybe get a little bit of discussion, although I realize that this Teams format is not ideal for that, but hopefully we'll be able to have some discussion at that point in time. Dwayne, do you want to pick up and talk a little bit about Research Day for the coming year?
Hey there, this is Dwayne with the EPI Research Day team, and this is a quick demo of what your Research Day conference will look like. This year's conference will be held virtually inside of Microsoft Teams. Microsoft Teams is a collaboration and communications platform, but we'll be using it as your conference's virtual venue. This means that the conference will be easy to navigate, and all of the information that has been created and generated throughout the conference will all be held inside of a single place, making it easy to find. To get Microsoft Teams on your computer, head over to aka.ms/getteams. Log in with your UF GatorLink credentials after installing the app. Now that we're inside of Teams, let's take a look at a few of the rooms inside your virtual venue. You can see here that you start at the general.
Can you get this
Think
to
this
show
as the lobby for the conference, where you'll find announcements and important information about events throughout the conference. Here, you'll also find links to join the keynote sessions from our two keynote speakers, Professor Andy Dobson from the Princeton University and Professor Philippe Sensonetti from College de France. Moving down, you'll see the
Slight technical issues here. Hopefully,
It is full screen for the audience.
okay, go ahead. Dwayne, we can't hear you, and it actually is not full screen for the
This will
audience.
be easy to navigate, and all of the information that has been created and generated throughout the conference will all be held inside of a single place, making it easy to find. To get Microsoft Teams on your computer, head over to aka.ms/getteams and log in with your UF GatorLink credentials after installing the app. Now that we're inside of Teams, let's take a look at a few of the rooms inside your virtual venue. You can see here that you'll start in the general room. Think this as the lobby for the conference, where you'll find announcements, important information about events throughout the conference. Here, you'll also find links to join the keynote sessions from our two keynote speakers, Professor Andy Dobson from the Princeton University and Professor Philippe Censonetti from College de France. Moving down, you'll see that there's a lounge. This is a place where you can connect with your peers, catch up with colleagues, post cat memes and GIFs, and start instant meetings between sessions or even after hours to have some fun. We have a tech support channel. If you're having any problems, please head here and post a message, and our help desk will be on standby to offer assistance. Now you'll see that we have rooms dedicated to each topic for posters. Inside these rooms, you'll find tabs at the top of the room that let you view posters in various ways. First, we have a virtual poster space. This is an interactive room that lets you view posters in an immersive way. Simply click and hold your mouse to rotate around the room, and then click on a poster to interact with it. If you'd like to view the PDF file directly, click the link icon at the bottom after selecting a poster. Next, we have a poster page. If you'd like to view your posters all on one page in a thumbnail view, you can do that here. Simply click on a poster if you'd like to view it directly. Each author will have a dedicated room where they can post content about their poster, as well as a meeting space so they can meet with folks who are interested in meeting the author. There will be a poster index posted in the general channel with a summary of each poster and a hyperlink directly to that poster's room. So there's no need to remember that you want to go back and revisit poster room 062. We'll be providing hyperlinks directly to each room for you. Thanks for watching this video. We hope that you found it useful. Stay tuned. There will be more detailed tutorials coming out in the near future. We look forward to seeing you at EPI Research Day 2021.
Okay, Dwayne, thank you very much. Further details to follow. And, you know, I think the Microsoft platform is an interesting one. I think it should be kind of cool to see what we can do. I would strongly encourage everybody, if you haven't submitted an abstract, please do so. We'd like to get as good a representation as possible. And again, obviously there's going to be a lot of COVID, but a lot of other things as well to keep folks up to date on what's going on. I think at this point in time, we're going to try to see if we can get all of the various individuals who were, you know, presenting some data and see if there are questions or comments. I realize the time is getting late and the format is not necessarily optimal, but nonetheless, we would welcome if, I know there's been some good ongoing discussions within the question and answer area, but if folks have got specific questions at this point or comments, let us know and we'll see what we can do to address the questions and go from there. Further questions? Thoughts about generally where we are with EPI. And again, I would encourage you, if this may well be stuff that we'll want to talk about in a side conversation somewhere in the corridor. But what do folks think?
Yeah, I just have a general comment. Looking at the types of work that all these different components that EPI is doing, like the stuff Marco outlined with all the phylogenetics and transmission analysis, and Derek with his modeling, and all the contact tracing data and transmission data. The vaccine work, the work John is doing on the virus itself. I think we would benefit from some, working together more closely than we have been, because there's a lot of currents running through, and there are other presentations too that I think there's a lot of currents running through these research. I think we could benefit quite a bit from working together more closely than we have. I don't know how we can bring that about, but I was very impressed, and some of us are doing the same thing with different collaborators on different levels. I think it'd be important if that we have some way to communicate and work together better than we have been. Just a general comment, but I don't think in some aspects the UF has been at the forefront, let's say, of COVID research, for example. And we certainly have tremendous talent and a lot of interesting things going on. But what we seem to have lacked is some common core way to work together. And I think the EPI provides that structure. So I know it's easy to talk. I know we're all really busy and really focused on different things, but it's just, since we're all sitting here together, I would like to see us be able to work together more closely. Yeah.
Can I quickly second that, because I had exactly the same feeling, and I really, I myself found out just today that there are so many other interesting things. I knew of some of them, but not all of them. And I really think that we really have sufficient critical mass to put together like a large program project. And there will be hopefully soon more money coming to NIH. The pay lines just went up. There is certainly a big interest in developing strong program projects in COVID and respiratory viruses, with the genomic capabilities, the exceptional modeling that Ira is doing and Derek is doing, biological expertise of John, and Mike with his amazing work in engaging communities, collecting samples, doing the test tracing. I personally planning to apply for at least one large extramural grant to fund the genomic consortium. But I think really we should, maybe within the next few weeks, even just the people here on the screen, get together for a brainstorming or planning team, because I really feel that there are all these different expertise, and EPI is the perfect container. What is missing is like the ability to kind of coalesce all of us in one single goal. I think there are tremendous opportunity for research, for making UF really on the front line of this, and for leveraging really substantial amount of funding from funding agencies. So I just launching this idea here. I'll be more than happy to send a doodle poll and put together a preliminary agenda if everybody agrees. But I mean, this was great because I think at least now we kind of know more or less what the others are doing and what each one can potentially bring to the table. But I think it would be the perfect time. I'd like to hear also what Glenn thinks about this, but it could be really the perfect time to kind of figure out a much larger project and contacting even NIH officials to see whether or not they are interested, that really involved all the different key components in which each one of us is doing so well right now.
This is Glenn. I'm in complete agreement. Any other thoughts from the panelists or folks in the audience?
Well, just again, sorry, pushing that idea Marco just mentioned, Glenn, you think it's possible to go after some kind of large center grant in emerging, infectious diseases? It would, or would this be getting in the way of the EPI's own charge? Yeah.
No, I think that's absolutely where we need to be headed.
Yeah. Okay. I think we should do it somehow.
I will give you a call next week, so we can start talking more in detail. No, seriously.
Yeah.
And then, let's involve everybody else here if whoever is interested, because I really believe there is tremendous opportunity here.
Yeah.
I think one of the things that is very frustrating is the inability to do a lot of animal modeling here. And that's a real drawback. If we could convince our animal care people to be more flexible, I think there are many things that are problematic here. One is the lack of proper caging, right? We don't have a very coordinated way of doing pathology work. And with regard to the animal studies. And for example, with a lot of the aerosol stuff I do, I'm able to collaborate with people in other universities very easily. And in fact, there are people calling me up today to talk about a ferret model, but I can't do it here.
I wouldn't go into about the mice modeling, for example, because that's not at all my field of expertise. However, in terms of animal models in general, my lab has been running on now for 10 years, an NIH grant on the SIV macaque model to study HIV infection. Now, macaques, of course, are very expensive. We don't have a primate facility here. But just to say that this is doable, we have a really long-standing collaboration with the Tulane Primate Center. They have all the animals or the expertise to do exactly what we want them to do, send us the tissues, we do all the downstream analysis. So at least in terms of large primate models, and there are many things that could be developed, especially for coronavirus with a large primate model, there is always the possibility, and we have already pretty strong ties and contacts to work with a primate facility. It has been working very well, even logistically speaking. So I don't think we need necessarily to have primates on campus to do that. Of course, for other stuff like the stuff that John was doing, that's different. But again, in terms of putting together a large multidisciplinary group, I think that we have the possibility to leverage primate centers that we have been working with and would be more than willing to work with an institute like EPI if we want to go in that direction.
Other thoughts and ideas, either through the question center or for the panelists?
One thing, this is Derek Cummings here. One thing that I've seen other institutions do a bit better is to coordinate at multiple stages of EPI studies, trials, design and sharing of samples, and because everybody is scrambling for power in any sort of statistical analysis. And so I think this has certainly been very helpful to me. I know about some of the things that have been going on, but not all. And certainly where, having more and longer discussions like this, where people can compare notes and see, opportunities and have more opportunity to kind of combine studies, share samples, and coordinate upstream, I think is key, because, later on there's some opportunities to do things, but often it's at that design phase where you can sort of bring things together and coordinate strengths across the institute and university.
So I've said this a couple times, this is Mo. Some things that slowed us down was the fact that we had a low prevalence in Alachua. But now the same things that have maybe slowed us down with getting samples locally is our strength, with the fact that we keep bringing students together and we are getting upticks in activity. In addition, we're now into the vaccine phase, vaccinating people. And a lot of the work is being done with the backdrop of high background infection in cities, in the larger universities that are associated with cities. And I've been kind of pounding on my high chair for about a month now that, pivoting to some really good vaccine studies here in the local community, this is our opportunity, because we have a relatively naive population. We really have, I've done 3,000 ELISAs, and we really have a very low seroprevalence because we have a fairly well-educated group of people, and Alachua County has had really great guidelines compared to the rest of the state. So I would really push all you really good study designers and those that are part of the community to really think about getting together and doing some nice studies that can look in a very clean way at antibody responses and duration of immunity that I think a lot of communities are not going to be able to do.
Mm-hmm. Let me just mention, looking at some of the questions and comments coming in from the audience. Again, Bob Cook's comment, we are currently recruiting epidemiology faculty. It's a great time to apply. Know anybody? So keep that in mind. Also from Bob, I think there's still potential interest in a T32 related to emerging infectious diseases that could include epidemiology. So again, something to think about as we move forward. Again, Fred Southwick has got a couple of comments on here. Again, emphasizing the importance of coming together and taking advantage of the wide range of expertise that we've got present. Any other comments? I think clearly there's the sense that, given what we showed today, there's the sense we need to try to facilitate the, bringing together some of these research areas and groups and thinking through long-term plans. Clearly, it's something we need to work on. What I would ask is that folks email me directly with ideas. Let me see what I can do to begin to facilitate some of this. Again, Marco, Ira, the others, clearly let's start talking about possibilities. But let's see where we can go with some of it, because I think there are some exciting possibilities moving forward. We are basically hitting the time frame where I promised we would, end things. I think what I would like to do at this point is to sort of, call a halt. Thanks to everybody who presented. I think it is a very impressive outlay in terms of the areas in which we've got work going and the possibilities for collaboration. I think I would strongly encourage people who are online to get in touch with faculty who may have presented today, or to see if we can work together to facilitate the ongoing collaborations that we've got within EPI. So my thanks to everybody. Have a great rest of the afternoon and evening. And I think we'll probably, hopefully get some of these groups together fairly rapidly and potentially put together a town hall again fairly soon to talk about where we've gone and where we may be going. And don't forget Research Day. It's going to be an interesting virtual experiment. but we definitely would like to try to get as many people as possible involved in our activities there. So thanks a lot, and have a good evening.
Thank you. Have a good weekend, everybody.
Bye-bye.
All, bye.